Hi, my name is Swin Lee. I'm from University of Washington. So today I'm going to talk about machine learning techniques focused on improving interpretability. And then I chose precision medicine as just an application area for these techniques because interpretability is extremely important in this space. But the machine learning methods I'm about to talk about are widely applicable to various other problems, some of which you are currently working on in biology, even outside of the biology. So I have like 60 slides to go through. I really want to be able to go through all of them so that you won't miss any of the things that might be helpful for your research. So let's focus. Okay, so first of all, what is precision medicine? So precision medicine refers to medical care designed to optimize efficiency or therapeutic benefit for particular groups of patients, especially by using genetic or molecular profiling. And then there are three problems which genomics or machine learning researchers, so basically everyone in this room, need to address to actually advance this field. So first, biomarker detection, identification, and second, disease subtype discovery from molecular data, and finally, prediction model construction based on EMR data. And then I'm going to show you right in the next slide why interpretability is so important in this area. And it's something the current machine learning methods cannot handle very well. Okay, so in any problem in precision medicine, and some of the biological problems you're trying to solve, you're given a set of features, patient features from various kinds of data. Sometimes you have high-throughput molecular data, such as gene expression data or genetic data. And a machine learning model would take this data as input and then provide a prediction of the patient outcome or a clinical phenotype or a general phenotype. And the prediction itself can be very accurate. We have a lot of very good prediction models these days in the field of machine learning. But a prediction alone doesn't yield sufficient information to take important clinical actions. So clinicians need to know why this model made this prediction, and the patient, of course, needs to know why. But what we can do with an existing set of tools is to just say it's AI magic and convince them to trust the results. So an important missing piece here is the explanations of the model and the prediction results as well. So for instance, why the features selected by your machine learning model make sense in terms of the molecular basis or biology or pathophysiology of the clinical event you are predicting or clinical phenotype. And which features contribute to the prediction and then how. And then these explanations can help clinicians take appropriate clinical actions. So this led me to identify the following two problems. So first, learning interpretable feature representation based on our prior knowledge. So we want to pre-select or learn features to begin with, especially when you use high-throughput molecular data as some of the input features, such that these features are likely to give some meaningful explanation of the outcome in an unsupervised manner. So second, making interpretable prediction from complex or black box model by estimating the importance of each feature on a particular prediction, so particular patient, so we can reveal individualized risk factor or individualized marker and their interactions. And they can hopefully engender more trust on the prediction model and then help us reveal novel risk factors. So in this talk, I'm going to show you 3 examples of how these interpretable machine learning can potentially transform several important areas of medicine. And I'm going to introduce some of the existing approaches as well, combination of my approach and then existing approaches. So first, molecular marker discovery problem. For precision cancer treatment, we show that learning interpretable feature representation actually help us identify robust molecular markers for drug sensitivity phenotype in cancer. Second, real-time prediction of various adverse events during surgery. And we show that making interpretable prediction help anesthesiologists predict hypoxemia event much better. And then last, individualized risk factors for chronic kidney disease. So the first part is about learning the interpretable feature representation. The second part is about making interpretable prediction. Okay, so the precision oncology. So say that there is a patient with AML. It's very aggressive blood cancer, and like other cancers, there are many anti-cancer drugs this patient can be treated with. The drugs that are designed to kill fast-growing cells. Cancer cells are, of course, fast-growing cells. And but standard therapy is not personalized in that every patient with AML is going to be treated with a very small set of drugs that are shown to work well in clinical trials. It's not individualized. So our long-term goal in this project is to build a machine learning-based system that takes molecular profiles of a patient and then predict the response to a large number of potentially usable anti-cancer drugs, and then gives us the explanation of the prediction and then its reasoning. So instead of just saying this patient is going to respond well to mitoxantrone, we also want the system to say that it's because the patient has a topoisomerase 2 activated. This drug is a topoisomerase 2 inhibitor. Okay, so that in the end we want to help clinicians to choose the best drug combination for a particular patient. So in this talk, I'm going to focus on a small portion of this project, which is on learning a robust prediction model from high throughput molecular data by learning interpretable features. So this problem seems actually really simple. I mean, it can be seen as a classic supervised learning problem. So how many of you are familiar with the supervised learning? Okay, excellent. So our collaborators in hematology got us training data, which consists of features variables X, here expression levels of nearly about 20,000 genes, and an outcome variables Y. In this case, it's in vitro drug sensitivity measures for 160 anti-cancer drugs. And then we have this data on a set of samples. So say that we have 200 primary AML patient specimens. So using this training data, we can learn a prediction model for each drug so that we can predict the drug sensitivity of a new patient based on his or her features. And to see whether this supervised learning approach works well, we first evaluated the simplest form of a supervised learning approach, which is a univariate model, which means that you first identify significant gene-drug associations, pairwise, and then we checked whether many of these pairwise associations are replicated in an independent left-out validation data set. And then we can compute the consistency rate defined as a number of associations replicated in an independent validation data set divided by the total number of significant associations, which can be interpreted as some empirical true discovery rate. So we actually measured this. We first ordered gene-drug pairs based on the degrees of association, so basically p-values, and then top some number of pairs and then measured the consistency rate, and got a really depressing result. Only 18% of them were replicated. But many of us who are working on this sort of analysis, genome-wide association analysis, may know that this is a very common problem, and for various reasons. First is high dimensionality. We simply don't have enough samples to ensure that the identified associations are not false positives. And the second, confounders. So say that there is a hidden variable that's a true marker for a certain drug. It can be some, artifact, batch effect, or it can be, like disease subtype, unobserved, undiscovered. And then let's say that in training data, it happens to be correlated to a bunch of genes. Then, these genes would have only indirect association with the drug. So these associations may not be real. May not be replicated. They are not true marker. Say that there is a true marker gene, which is a disease driver, and then it's a true marker for another drug, and then it has many downstream genes. So their expression levels are are correlated with each other. So it turns out that the association between these downstream genes and the drug are indirect. They may not be real. So, which is the reason that it's not replicated. So diseases heterogeneity also contributed to the problem. So there is a discrepancy between actual biological interactions and then the observed association between genes and drugs. Therefore, we cannot fully rely on these observed associations between genes and drugs. David?
So I completely agree as you assume the correlation, but it should still in principle replicate, right? Because unless you for some reason don't expect to see that confounding validation set.
Yeah. So it depends on what the validation data set is, right? It could be from the same lab. We consider the 3 validation data sets. One, other publicly available data. The other one is, the data obtained in different dates from my collaborator's lab and so on. Yeah, good point. Okay, so an alternative solution then is not pure supervised approach. Instead, we want to do some kind of unsupervised learning. In particular, we want to do unsupervised feature learning. So how it works is that then we first integrated a publicly available high-throughput data set from many AML patients. And then we try to extract a small subset of genes likely to represent important disease process events in an unsupervised manner. So for instance, and then reduce the dimensionality of gene expression data into smaller learned features. So one example is that it's an expression level of a hub gene, the genes that are densely connected with many other genes, infer the gene regulatory network, and Sushimura gave very excellent review on the gene regulatory network reconstruction. You can extract those genes and then you use only those genes, or use the average expression of a module, or take only the genes that are cis-regulated by each mutation or methylation, okay? So, and then use only those selected genes for learning, for solving this supervised learning approach. So why did I just talk about many network-based features? Because from our previous studies, we showed that simple network topology features such as hubs or modules, they actually helped identifying robust expression markers for important cancer phenotypes. And then it actually provided a good way to reduce the dimensionality of gene expression levels. So for instance, we developed the INSPIRE method to learn a conserved module network based on multiple expression data sets from different studies. So different microarray platforms, some of them can be RNA-seq. And then we linked latent variables, each representing a module, to a clinical variables. And then, so we can better characterize the module and then identify markers inside the module for these clinical variables. And then the result of this study was that it led to the identification of an interesting gene, HAPX. It's a regulator in one of these modules as a potential driver for tumor-associated stroma. It was highly expressed in true stroma and the mesenchymal stem cells. And then for resectability phenotype, so how much residual tumor left after surgery. So the implication then is that HAPX can be a therapeutic target in neoadjuvant therapy. So you want to shrink the tumor before surgery, okay? So this work was actually, excited at least one person in this room. I'm not sure if he's here. Casey, right after it was published in Genome Medicine, he wrote about our paper and as editor's choice in Science Translational Medicine. Is he here? Are you here? Yeah. Okay. So similarly, we developed the discern method to identify genes whose neighbors are significantly different between two conditions, so cancer and then normal tissues. And then we showed that these small set of genes selected based on this criteria provided robust features for survivor prediction model in 3 different human cancers: leukemia, breast cancer, and lung cancer. And then we also showed that hubs inferred purely based on gene expression data indicated important cancer mutational driver event. So all these results show that simple network topology-based features, such as hubs or modules, they help identified in an unsupervised manner. So don't look at the phenotype data, just based on gene expression data. And then they provided a robust feature selection methods for important cancer phenotypes. Now then, back to this drug sensitivity prediction problem. So here the difference is that we want to be able to combine multiple pieces of such information, not just focusing on hubs or modules, but multiple things. So, so how does it work then? So we modeled the prior distribution over the weight of the contribution of particular gene G on drug D as a normal G distribution with a mean 0 and then sigma, certain sigma. And then this sigma value determines how much we encourage the W to be deviated from 0. It's a prior distribution. If we believe that some gene does not have much potential to be a biomarker for drug sensitivity, then this gene is going to have a small sigma, whereas some genes that we believe to have, have high potential to be biomarker for drug sensitivity phenotype will have a high sigma. So, you really don't want to, fix this, set this sigma value for each of the 20,000 genes. So instead, we want to learn these sigma values from data based on various properties of individual genes in terms of how much potential it has to drive the disease progression. So the genes that are, like high-level decision makers or regulators in the disease, those genes are more likely to be a good indicator for whether certain therapy, is going to work or not. Right. So what features did we consider? So this is cancer. Can anyone give me any answer? What kind of gene features would be important here? It's cancer. Somatic mutation. So how much the gene is mutated in AML patients? We can incorporate lots of like TCGA, AML sequencing data. Right, and then we also consider the homeness feature, whether how many neighbors it has in the gene expression network constructed based on some regulatory network inference algorithm. We also consider the gene annotation database. So whether the gene is known to regulate other genes, transcription factors, chromatin molecules, and translation initiation genes, and so on. We also consider the genomic CNV feature, and then an indicator whether how frequently the gene is methylated in AML. So we called these features merge feature, and then merge algorithm. Okay, we want to merge all this information to better select the molecular markers. So our merge algorithm can simultaneously learn the weight of these features V, because you do not know which ones are important by the extent. You don't know how much weight you have to give to each of these merge features. So we want to learn this V as well as this W. So you want to optimize some joint objective function. So a simple approach without this part, you can just it can be viewed as just pairwise correlation calculation. It's a univariate regression model, whereas if you use a merge algorithm, you have two additional terms. So one of them is the weighted L2 regularization term, where the regularization parameter for each gene is specified based on each merge feature, linear combination of each merge features. Okay, so the high-level intuition of this algorithm is that you want to project very high-dimensional gene-drug associations, 20,000 genes, 160 drugs into lower dimensional space generated by your prior knowledge encoded in this merge features. In other words, you want to believe this gene-drug association only when it is supported by any of these genes driver features, merge features. Okay, so this is a way to make your associations more interpretable, explainable. Okay, so the result was that it outperformed its competitors. We compared with, from some simple approach or some more sophisticated approach, including one that won the DREAM competition for drug sensitivity competition, and then measured the consistency rate, which you just saw, and then we showed that the merge does better than other methods. We considered the 3 test data sets, and then we used a number of other evaluation metrics such as actual prediction rate and so on. So you can see this in our paper if you are interested in what results we. So quickly moving to the next piece of result, and then the question is what exactly are gene-drug pairs that are new, and then something you want to actually experimentally prove. So we found many interesting gene-drug pairs that make sense based on known AML biology, but one of them was new. So which is that the SMARCA4 expression associated with increased sensitivity of these 2 drugs. this finding is clinically really important because these two drugs are actually used in today's AML treatment. So identifying molecular markers for these drugs is going to have can have immediate impact. And then I just want to note that the SMARCA4 would not have been captured if your analysis was purely based on supervised approach. They have a significant p-value, but it's not, like very much top listed enough to just, jump to perform experimental validation. Okay. So, this motivated us to do experimental validation in my collaborators' wet lab. We picked the AML cell line that showed low SMARCA4 expression and then transfected that cell line to overexpress SMARCA4. And then measured drug sensitivity. You can see that when you overexpress SMARCA4, the cell becomes a lot more sensitive. This is a classic, the dose response curve where x-axis is drug concentration, y-axis is cell viability. You can see that the transfected cell line became a lot more sensitive to this drug and the other drug as well. To be sure about this finding, we picked another cell line that already showed high SMARCA4 expression and overexpresses SMARCA4. In that case, there's no change in drug sensitivity. Okay, so we are trying to apply. So this, merge, we characterize it as a probabilistic model-based unsupervised learning approach, and then we are trying a slightly different version of the unsupervised feature learning approach, which is that, changing this part into some kind of a deep learning approach. So. So we use all many gene expression data sets from AML patients and apply variational autoencoder and then use these latent variables. We tried many numbers and then found that 10 is a good number in terms of the prediction performance. And then we use these learned features to predict drug sensitivity. And then again, this has been done before. We are not the first people who did it. So Casey here applied the VAE to 10,000 TCGA pan-cancer data. In this case, the phenotype is not drug sensitivity. It's other, important cancer phenotypes. Okay, so my first-year student collected, where's her photo? So this first-year student, Aisha, collected all available AML gene expression samples from the most popular Affymetrix platform, so it's this number, AML, and then leukemia, and then we applied the VAE and then extracted 10 latent variables, and then we used the VAE with the 4 hidden layers, and then we used the lasso to predict drug sensitivity, and then here is the live-on out-cross validation test result across all 160 drugs. You can see that when you use all genes versus when you use a VAE, you see you see 10% error reduction when you consider only small subset of drugs that are actually widely used in cancer, and then FDA-approved drugs used in AML. Overall, these methods do better, and then especially this VAE-based method shows a lot, higher error reduction. Okay. So these very preliminary results are very promising, and then we are trying to, gather more data to see whether that affects the performance. So this was the first part of the talk. So I'm quickly moving to the second part of which is completely different machine learning approaches. So this second part of the talk is about our machine learning system that can make real-time predictions of various adverse conditions, adverse events during surgery, and then can explain why the event, is likely or to occur. So surgery is an integral part of healthcare, and then but still, surgery and anesthesia pose a considerable risk of complications and patient death. So this creates great need for data science to utilize this perioperative data before, during and after surgery data to positively impact surgical and anesthesia care. So in this project, we focused on unwanted physiological condition called hypoxemia, meaning low blood oxygen level that causes a serious patient harm. So if hypoxemia can be predicted or anticipated before it occurs, even a few minutes before, then actions can be taken by anesthesiologists. and then proactively, to proactively prevent hypoxemia and then related patient harm. So to do that, we developed the Prescience system. It's a machine learning-based system that can predict the near-term risk of hypoxemia in the next 5 minutes based on various data during surgery, and then explains why the patient or surgery-specific factors that led to that risk. So here's how it works. So based on about 20 static features, so the data from patient charts, and then 45 dynamic features, what you measure, the patient's vital signs and so on. Based on these features, the Prescience can make real-time prediction in the form of a numeric representation of a risk. So for instance, a 2.5 odds ratio. And then also, its major feature is that it can explain why this prediction was made. So, regardless of the complexity of the machine learning model used to make this prediction. So, this Prescience model was trained based on 50,000 surgeries in the AIMS data, which consists of high-fidelity real-time data, such as the data from patient monitors and anesthesia machines, and input-output fluid totals, lab results, and so on. We also used the static data, such as ASA code, surgical procedure, diagnostic codes, and then patient demographic information. And then we extracted 3,900 features based on these variables. And then we trained the gradient boosting machine model, GBM model. I'm going to explain to you why we chose this model later. And then ensure that there's no bias towards the final test result. The test data was initially compressed, it's zipped, and then left untouched until the development is done. Okay, so now, so we are ready with this training data, and then my PhD student, Scott Lernberg, so he learned a prediction model and then measured its prediction performance on new held-out data, and then drew this ROC curves from 10-fold cross-validation test. And then two best performing models are one simple model, which is a linear model, which is perfectly interpretable. The weights will tell you which features are important. And then the ensemble tree model, which represents a complex model, which is often considered to be a black box. And then, actually, this ensemble tree model is significantly better than the linear model if you actually read these numbers, in the y-axis. And then, many of the machine learning researchers are actually familiar with this situation, the trade-off, the tension between accuracy and interpretability. So with the large modern data sets, the best accuracy is often achieved by complex models. Even experts struggle to interpret. And then, linear model is really easy to interpret, but it often works worse in terms of the prediction accuracy. So to resolve this challenge, we developed the machine learning techniques to estimate each feature's importance for a particular prediction made on a particular patient at a particular time point. You focus on just one sample and then estimate the importance of each of these features, so phi 1 through phi p, and then give and then these, phi values are explanations for the prediction made. Okay, so, we claim that our ability to provide simple explanations of predictions from arbitrarily complex model eliminates the typical, this trade-off, accuracy and interpretability trade-off. And then we believe that it allows broader applicability of machine learning models to medicine. Okay, so how does it work then? How exactly can we compute this phi value? So to show you how exactly we compute this, let's consider a simple, more intuitive example. Not many of us should be expert in anesthesiology. Okay. So this is a John, a typical bank customer, and then like many other customers, when he applies for a loan, his information is sent through some prediction model. Okay, and then this model is designed to calculate the risk that John will have a repayment problem which unfortunately for John, it is 55%. And because his risk is high, the bank declines his loan application. So natural first question then is why? And then the bank also want to know why, because this is a key business decision. But unfortunately for the data scientist that built this model, she used a complex model. So these questions are hard to answer. So it's again, AI magic. We want to do better than this. So how do we explain then the prediction of a potentially complex black box model? So one approach is to change the model input. Where X1, for instance, could be, for example, represent John's age, and then see how the model behaves. So by hiding such a feature from the model, or a set of features from the model, we can learn about how features affect the model's output. Okay? So if we want to summarize these effects, then we can attribute an importance value to each feature, these phi values, and then we can replace the model with a simple sum. So these feature attributions approximate the effect of hiding features from the original model. Okay, makes sense, right? That's the importance of each feature. So note that these attributions are specific to the model F. Okay, the prediction model complex being explained, and also X, the input data, the instance, particular bank customer and his or her data, okay, used for this prediction. So that's why I said phi 1 and then it's F and X. Okay. So it turns out that this way of explaining a model's prediction as a sum of feature attributions is widely used. In fact, there are at least 7 different explanation methods that all use a sum of feature attributions to explain complex model output. So LIME, for instance, is a popular method. It's designed to explain model predictions by fitting a simple model to the original complex model locally. So where this locally means near the current set of inputs we are explaining, so particular sample, okay? And this simple model they use is a linear model of toggled inputs, okay? And so it can be viewed as a sum of feature attributions. There are 3 methods based on ideas from 1950s game theory about allocating credit. And then they also produce a sum of feature attributions, so it can be in this class. And all of these methods can explain the output of any black box model. They are model agnostic. However, there are also methods designed to interpret output of specific types of complex models. So for instance, past explanations is targeted at tree ensemble model, like gradient boosting, while deep lift and the relevant propagation target neural networks. Deep lift is from Anshu. Is Anshu here? Oh, no, he's not here. Okay, anyway, so since these methods all produce explanations of the same form, they can be considered part of the same class. So we defined these methods we defined the class called Additive Feature Attribution Methods class, AFAM, okay? So, and then now, noting this previously unappreciated unity among explanation methods is helpful. But even more interesting here is that a connection between game theory, 1950s game theory, implies there is only one way to assign feature attributions. So only one set of phi values while maintaining 2 important natural properties, okay? And then what are these properties then? So, so the first is local accuracy, so which states that the sum of the feature attributions equals the original model predictions. So most of these methods, 7 methods, have this property, okay? And the second is a property we called consistency, which states that if toggling a feature in one model makes a larger difference than toggling the same feature in another model. See, notice this model changes. So you toggle this feature in model F, and then you try to toggling again with a different model F1, then the importance of the feature in the first model should be larger than in the second. Okay, so this sounds, this may sound that it's not very important, but in fact, it's a really important feature. What this means is that if a feature attribution method is not consistent, it can actually lower a feature's assigned importance when the true impact of that feature actually increases. So this is actually a fundamental problem that casts doubt on any kind of a comparison between features even in one model. Okay? So under these two conditions, these two properties, the feature attribution values are forced to be the classic Shapley values from this old game theory. Okay, there's a proof that says there's a specific set of phi values that satisfy, only one set of phi values that satisfy these two, that are compliant with these two properties. So now a number of current methods have not previously considered these properties and then the uniqueness result they imply. Okay, so what that means is that it has important implication for each of them. So in contrast, these three methods already had connected with game theory are compliant with these two properties, but they are very inefficient, much less efficient to compute than these other methods. So we proposed a unification of these approaches that leverages the unique strengths of each. And this unified approach presents a set of ideal values we called SHAP value. That can be, that can then be approximated by different methods depending on the prediction model F. Okay. So then, how exactly can we compute these values? And then, first of all, what exactly does these values mean? Okay. So to see, let's return to John and answer why his loan application was denied. So to explain this denial, it is important to start with the base rate of loan repayment problem, denoted here, by the expected value of the model's output. So to explain John's risk, which is 55%, we need to explain how we got from the base rate to his rate of 55%. So how did we get there? So that's how the prediction occurred, right? So to do this, we start at the expected value of the model output and then condition on a single feature, John's age first. So since this increases, John's risk by 15%, we attribute this, this much to this John's age feature. And next we condition on John's occupation, which is very unstable, and again attribute the change in expected model output to his occupation as a day trader. So if we repeat this for his open accounts and then marital status, we end up conditioning on all the features, and so we arrived at 55%, John's predicted risk. And it's important that in this process, the order matters, unless it's a linear model. So for instance, the impact of John being a day trader, it could be particularly bad if you are a young day trader. It means that the impact of a day trader could be larger if you already know that he's young. So which means that if we reverse the order between, if we introduced this feature first, day trader first, and then age, the day trader could shrink, okay? So these SHAP values result from averaging over all n factorial possible orderings. And then, of course, this is not how we want to actually compute these values. So we need to develop actual learning method to estimate or compute the exact SHAP values. And then one of the papers you discussed, some of you discussed in the journal club, was about how to efficiently compute the exact SHAP values in ensemble tree model. Okay. So let's now return to Prescience. We are getting really close to my favorite result in this talk. So Prescience outputs the risk prediction and its explanation, which shows a set of features that increase the risk of the purple-colored features and then decrease the risk of green-colored features. And then the plot at the bottom here is equivalent to rotating this feature explanation bar by 90 degrees and then stacking these explanations for each time point horizontally. So you can see these real-time feature explanations. Okay? So that's what we hope the anesthesiologists see when they are when they are in the OR. Okay, so to test the potential of a prescience to aid hypoxemia prediction, we replayed the pre-recorded surgery data from test sample procedures, which we did not use to train the model. We developed a web-based visualization system and then showed them to 5 practicing anesthesiologists. They are all professors. They are not, none of them is like resident or not. The residents are bad, but like these are really very experienced anesthesiologists. Okay, so we asked them to solve this quiz, go over each of the 260 cases, and then were asked to assign a relative risk of hypoxemia as compared to a normal acceptance rate. So there is a score from 1 to 9. There is a sliding bar, and then they can choose the score. So it took many months actually to have them, finish this, because these are really busy people. So we drew the ROC curve, and then it was averaged across 5 anesthesiologists. So here's the result. The prediction accuracy of anesthesiologists, green colored, markedly improved when the anesthesiologists were helped by Prescience risk prediction and its explanations. And then what this result suggests then is that if anesthesiologists currently anticipate 15% of the event, the y-axis of the ROC curve, then the Prescience assistants, they can anticipate 30% of these events. So what this actually means is that it's estimated additional 2.4 million surgeries in the US, and then large portion of which may be preventable because they are attributable to modifiable features. Okay.
So this is the 5-minute prediction?
After 5 minutes, yeah.
So do you look at the same case more than once then, or you only look at one
Yeah,
case?
this is a really good question, because the anesthesiologists solve the problem without and then with the Prescience aid, right? So to avoid that case, one anesthesiologist is working on both cases. We divide them into 2 groups, okay, A and then B group. A is going to solve the problem, just the same problem without Prescience help, and then B group, and then the same problem, in a different set of a problem, they solved with Prescience aid, and then it's reversed in the other group, and then we averaged them. And then I'm not sure if you can see this, but this variation, some of the variation comes from that, because A and B group, they have different sets of anesthesiologists. Okay, excellent question.
Whether the anesthesiologist, if they see the whole time,
Yeah.
the whole, an hour worth, right?
Yeah.
If an event happens in the first 10 minutes versus after 4 hours of surgery, is there a dependence there on the anesthesiologist's performance, do you know?
Wow, that's a very, if we were asked to revise the paper one more time, we will do that. Yeah, hopefully that's not going to happen. Hopefully this is the last time. Yeah, anyway, that's an excellent suggestion. Maybe for the next paper. Yeah. Okay, so I'm actually waiting for a question here, a particular question I'm thinking about. What if just, Prescience makes the prediction, right? Okay, so I separated into 2 slides because when I present this result to clinicians, I don't show this. Just joking. So interestingly, the prediction performance anesthesiology with Prescience explanations was slightly lower than direct predictions made by Prescience. So what this means then is that anesthesiologists adjust their risk estimate for patient, if they make that prediction away from what Prescience originally predicted, then they are more likely to be wrong than right. Okay, I'm not gonna add more than that. Yeah. All right, so the third, how am I doing on time? So the third part of it will be super fast. Okay, so I will briefly talk about our ongoing effort on CDK, chronic kidney disease. So CDK refers to a condition characterized by gradual loss of kidney function over time, and it can affect almost every part of your body. So because of that, a lot of people who have a CKD has a higher increased risk for cardiovascular events and progression to kidney failure. So one way to prevent the CKD is to manage, your medical condition with the doctor's help. Therefore, predicting in advance, a few years or even 10 years advance is going to be really important. Okay. So again, given a set of features, from various kinds of data from blood and urine, we want our computer to predict the outcome in terms of whether a particular patient is going to have ESRD, end-stage renal disease, or not. So ESRD means that it's a condition where life is sustained only by dialysis or kidney transplantation. Very bad condition. Okay, so if we use the SHaP method, we can actually revise our goal by making a prediction with explanations, so they can be interpreted as individualized markers, and that way we can define a new disease classification system based on these explanations. So people who have the same level of kidney failure risk, they can have different reasons, and they can classify it into different disease subtypes. We used the CREEK data that contain nearly 4,000 participants with the CKD who remained in long-term follow-up. So some of them many years, but the average was 6.3 years, and the standard deviation 2.4 years. And then there are some number, 760 ESRD cases. We used the Cox regression model with the GBM complex model, and then it actually worked better than just a linear version of the Cox regression model. So the CREEK data contain 335 features, which our expert collaborators categorized into 15 meaningful groups. So for instance, these 12 features are, they fall into the cholesterol category. And then this grouping was done by these experts, nephrologists and biostatisticians, not me. So we totally trusted this classification. So we developed an effective visualization of the global importance level of features across all individuals. Okay, so we saw these phi values specifically for individual patients, but you can sum them up to get a sense of which features are important globally. So here, we called it SHaP summary plot. So some of you who discussed our paper might have read about it. So if every individual in this data set is run through the model, and then a dot is created for each feature attribution value, these phi values. And so one person gets one dot on each phi values line. So each of these dots is one person. And then dots are colored by the features value for that person and then piled up vertically to show density. So this is like a violin plot, but we believe for this purposes it's better because for violin plots you have to use arbitrarily smoothing kernel widths. Okay, so. And then when we collapsed them into 15 meaningful categories of features, here is the result. And then our expert collaborators were really happy to see these two main features. They were expected to be important for CKD. What's important is the previously unknown risk factors. So we developed another way to visualize our SHaP value result, which we call the SHaP dependence plot. So it's alternative to partial dependence plot. And SHaP dependence plot uses the SHaP value of a feature for y-axis. So y-axis plots the SHaP value of a particular feature. And then x-axis is the feature's value. Okay, so it shows the relationship between feature value and then its predicted importance of that feature on the prediction. So by plotting these values for many individuals, so here again, each dot is an individual, we can see how the features attributed importance changes as its value varies in the x-axis. So while standard partial dependence plot only produce lines, SHaP dependence plots can capture vertical dispersion. What does that mean? Feature interaction. Okay, so the individuals who have exactly the same feature values have a difference in terms of their explanations to the risk, right? So it captures the feature interactions, and then these effects can be visualized by color, for instance. Then x-axis is the feature values of the main systolic blood pressure feature, and then y-axis this color means the feature value of another feature that's selected to have the highest interaction level with this systolic blood pressure feature. Okay, so for this particular feature, I just picked this because it's really interesting. Because our expert collaborators have found that it's really interesting. So for systolic blood pressure, an interesting cutoff point, the inflection point, is around 120 milliliters of mercury. Why is it interesting? Well, this is consistent with a study by Sprint research group, which showed that targeting a systolic blood pressure less than 120, rather than, as compared to the standard 140, resulted in lower rates of cardiovascular events and then deaths from any cause. Okay. So, and then we didn't use any of this information, but the SHAP plat picked 125 inflection point.
Could it be in your training set for some?
Yeah, so right. But, as I said, the standard care is cutoff is 140. Yeah, doctors don't act based on this threshold. Yeah. Okay, so the SHAP plat on the urinary albumin concentration is also interesting because the interesting inflection point was at 30, which is a medical definition of microalbuminuria. 3,300 were not really captured, but it's a medical definition for macroalbuminuria, right? So I'm going to quickly so this is the part that can be very useful for you. So we have a very easy to use implementation of SHAP, both for XGBoost model and for model agnostic version as well. So you can also generate the SHAP dependence plot. So in this case, this is the dramatic example that actually captures the feature interaction. So this data is slightly different. It's not nephrology data. It's actually another data set. So this prediction model is to predict the mortality, the survival time, based on 14 features from large number of subjects. And then it shows that depending on the age, so the systolic blood pressure matters, especially for young people rather than older people. So if you want to read more about this result, you can see this our paper in our archive. We also have a popular implementation of the integrated gradient method. It's not SHAP method, but it's widely used, and it was developed by Google researchers, published ICML paper. My student now, Hiranuma's implementation. So he implemented this method that developed by Google researchers, and then this implementation works for any keras model. So what this means is that if you have a keras deep learning sitting around, then you can just, you want to quickly explain it, then his package can do it in 2 lines. Okay. All right, so in this talk, I presented 2 new machine learning methods focused on improving interpretability. So, and then I showed that, I showed 3 examples applications of how they work. And then, so this set of people I want to acknowledge here. So the students in the Lee lab, PhD students from computer science, some of them are MD PhD students, and then statistics, and then a set of collaborators in genome sciences, the other department, and then various other clinical departments. It was funded by various funding sources. Thank you.